Today's presentation is going to be about the need for whole genome sequencing for outbreak analysis, particularly during foodborne infections, waterborne infections, and environmental related infections, how whole genome sequencing can help to solve such outbreak analysis. So the whole genome sequencing can be used for the pathogen detection, identification, and it can also be used to determine the drug susceptibility, which pathogens are susceptible to a particular antibiotic based on the sequence analysis, if there's any resistant genes found or if there are mutations in genes that can contribute to the drug being inactive. And also it is used for epidemiological typing, where we can know the particular strain identity, and if a particular strain is mutated and if it's causing infection in an area, and also if the strains causing infection are related with other strains in other geographical areas. So all such things come under the epidemiological typing. Moving on to a simple workflow is being explained here, and we can see that for such analysis, the first important step is the pre-treatment of the clinical sample. So in order for a sequencing project to successfully give us the results that we are looking for, the sample collection and storage plays an important role. So once the tissue or the nasopharyngeal um, uh, swabs or saliva swabs or blood samples are being collected from the patients, they have to be stored properly until the experiment starts. And another important factor is the presence of the host DNA, because we want to be sequencing the microbes in the sample, the microbial communities, and we do not need the host DNA. And since 90% of the DNA would be the host DNA, we need to deplete them or enrich for microbial DNA, so without affecting the bacterial DNA copy numbers. And how do we do it? There are a number of steps: enzymatic analysis, enzymatic lysis, chemical lysis, or mechanical lysis, disruption, and then we extract the DNA, following which we choose the sequencing method. So here I've mentioned the 16S ribosomal RNA sequencing, PCR-based sequencing, and the 18S ribosomal RNA PCR-based sequencing, or you can go for the whole shotgun metagenomics, which is has a higher resolution because it gives the whole all the microbes present, even the low abundant microbes present. And after the libraries are prepared, after the PCR and the amplification and the libraries are prepared, they're sent for sequencing. And once we get the reads, we can then match the reads or compare the reads against a nucleotide database such as in the NCBI, and we can run a blast to see if the um, reads that are matching with different communities, species, and then we can go ahead with the analysis. So as mentioned in the previous slide, the collection, the storage, and how the human host DNA is depleted plays a big role in the data that we finally get. So we know that an important enzyme being used is saponin. Saponin, along with nuclease such as DNase, can help to deplete the host DNA because the mammalian membranes are more easy to break rather than the bacterial, and also the circulating free uh, DNA can also be removed, and and a number of microbiome kits by Kiagen company also is suited for this particular study. So you can check out the various kits in the Kiagen websites that are for such studies. And in every sequencing project experiment, there are three steps, stages. So the data that we receive falls into three stages of analysis: the primary, the secondary, the tertiary. The primary and the secondary is for all sequencing experiments. It is followed in every analysis that is needed. And the first being the quality control and the read accuracy assessment. And if there are low quality reads found in our data, we can trim them. And followed by which the reads that we get have no order, and we need to assemble them, or we need to map them to a reference genome that's already available in a database that's being used by other people and approved by other people. So these can be followed, and then is the tertiary analysis. The tertiary analysis is where, depending on your project need, you can add or exclude these steps. So it includes annotation. So annotation is also very important because we need to know what these reads are and what are the genes, where are they located, and we need to know about that, and then followed by which we do the variant calling to know if there are the mutations, and then we do the AMR detection, where we want to find out if the particular pathogen is carrying some resistance gene, if it's acquired resistance, or also in the case of the variant calling, we can find out if there's a mutation in a particular gene that's causing a different protein or a dysfunctional protein. And then we also do in silico bacterial typing, and another important step in most of these studies is conducting phylogenomics, where we want to know where these pathogens cluster with other pathogens, with other strains that are being studied. And some other analysis include feature extraction, insertion sequence, because most often these bacteria can acquire a foreign segment from another pathogen by horizontal transmission, and also they can bear some virulence factors which cause them to cause more infection in the patient. And so all such analysis is key for outbreaks, for solving such outbreaks, and for making uh, clinical decisions, and to know how the pathogen, how the disease is spreading in the world, and to help for the making the policies for the public health. That's it for now. Thank you for watching, and stay tuned for more such videos.